welcome back to the Science Connections show, Nature, Curiosity, and Hope. This show is about exploring nature through science to discover the wonderful world around us, from the quarks to the galaxies and everything in between. Our guide will be our innate curiosity, asking critical questions that only lead to more questions, embarking on a journey of trial and error, failing many more times than succeeding, in the hope of creating a better life for humanity in harmony with the universe. In each episode, we will meet a scientist who will share the science and laws she or he have discovered. We will ask our guest scientist to tell us why she became a scientist and how her journey unfolded, sharing both the triumphs and the challenges she faced in her roller coaster journey of discovery. Lastly, we will learn how their scientific discoveries affect our everyday lives, and without them, life would never be the same. Today's episode, we welcome Dr. Hala Abou Assi from Duke University. Welcome, Dr. Hala. We're so excited to have you with us, and we're really looking forward to learn more about your science and your work. The title of this episode is "The Jiggling and Wiggling of Molecules." So we're gonna, get up and have a dance. So welcome, Hala. And by the way, Hala in Arabic means welcome. So let's begin. If you'd like to share with us, what got you into science in the first place? How did your journey start?
First, I'd like to thank you so much for the invitation. I'm excited to be here with you today. So as you mentioned, it's all about curiosity. I was a curious kid, and so I was very interested in chemistry because chemistry is everywhere around us. Everything that you eat, that the medicine you take. So it's like I'm always curious, why does my mom add the ingredients while she's baking a cake in a certain order? Why does she add the baking powder? How does it make the cake fluffy? This is just like one simple example. How is the animal waste helping plants, and why? Like these these farmers long ago didn't know how it's doing it, but they were chemists in their own way at the time because they knew that. That's why I got interested in all of these questions, like how do these reactions happen? Why do things change form? Why when you add one thing to the other, it will change color? And then, of course, another thing that got me more curious into the science is that most of my family members are most of them are medical doctors, but then when I joined and learned more chemistry, I preferred to be in the chemistry world and more in the science, not in the medical doctor. It was the curiosity of the child that made me want to learn more, and why.
So how did that curiosity unfold later? You graduated from school and you went to the university, but then what made you pursue a career in research rather than doing something else? What was it that you felt that calling? And tell us about your feelings, and maybe if you want to share some of the triumphs or challenges that you faced along the way.
Sure. So it actually was also in school where I really enjoyed chemistry and biology as majors. So I had really nice teachers that made the subject even more interesting. So that they also have some credit, because otherwise I would have hated the chemistry or biology at the time. So so during either high school or even before that, I had good science teachers that made it an interesting subject. And then when I joined AUB, I joined as a chemistry major and loved running experiments in the lab. We had a course where they give you an unknown, where you want to, by the end of the semester, figure out what is the unknown that you were given at the beginning of the semester. I think that was my the best lab I did, because it was every day you go in there. You had the freedom to go whenever you want during the lab the regular, when there's like lab technicians that can be around in case you blow up something. But so I really was going there enjoying like what will I learn more today about this unknown that I have. So so I enjoyed this part of it, like solving for what's not known. So after AUB, I went to McGill and I joined Professor Masad Dama's lab. I think the shift from AUB to McGill was the most challenging. And the beginning was super tough because, traveling at such a young age and leaving family and, all your loved ones. And Lebanon were like so family oriented. So, all my relatives and cousins are all like in the same building or like building across, and to go out of all of this to a totally different country, that was very, very hard, very hard. And also like the level, like moving from, although AUB is like a great university, and I just moved from an undergrad straight to PhD, that was also another challenge. So the beginning was very hard, but definitely was worth it.
So can you share with us a story of where you faced the challenge and how you dealt with it, and came out on the other side, more or less, let's say, older, more mature
Sure.
from that experience?
Yes, for sure. The move, as I mentioned, was hard. Of course, I had some a friend that was already in Montreal who helped me, or family friends who picked me up from the airport, helped me set up my room at the dorms. So I'm very thankful for the people that helped me along the way, for sure. But then when I went to these classes and starting my PhD in a lab where everyone, you see everyone. At the time, you think like everyone knows so much and they're much better than you, but you're just starting new. So of course they will know more than you, right? But to me, it's like, oh my God, like I don't know anything, and this is very hard. And also the courses that I had to take, the courses were like graduate level courses that, for most of them, I unfortunately didn't have the prerequisites, because like here undergrad, like they took, for example, organic 3, which was more about nucleic acid chemistry, and but we didn't have that at AUB, or at least it wasn't required. So at the time I didn't take it. So when I joined these classes, I found everyone around me, even the undergrads, participating and knowing everything, and I'm sitting around like, this is way beyond me. So I had to put so much extra effort, so much. That's why it was very hard. So, but I was like, okay, I took this decision. I am here. I have to do it. So I like tried to, get the books or the lectures that can help me fulfill any gap I found like I'm missing, or a prerequisite that I have to take. So, because I'm already taking the course, so I can't like go and take the prerequisite, but I can on my own like time try to get up to speed and learn it, which required way more hours than other people that would in the class with me, for example. Again, I will have to admit that during the first 4 or 5 months, I was about to quit every single day. I call my sister and be like, can you book me a ticket? I just want to leave. It's too hard. Like, I can't keep up. And she will be like, just try one more week. Let's try one more week. And then it ended up being great. And I enjoyed doing the work afterwards. I did all the courses. Maybe I didn't get A+ on all of them, but, I passed all of them. And then I ended up finishing my PhD with really good, I had several first author papers. I won the Best Thesis Award of the year in the department. So I think the challenge was, totally worth it. And I'm very happy for the outcome.
So you keep mentioning the word hard. And I just want to go back to that. That feeling of things are hard, yet you stuck it out, you persisted, and you kept going, and you had multiple first author papers, you won an award. So tell me, how did you bridge the hard to that kind of success? What was that bridge like? And I'm asking you in context, if you were looking back and talking to yourself though, all those years back, what would you have advised yourself on building that bridge from hard to successful?
I would advise myself to calm down. Things are not as hard as I thought they were at the time. And, like that exam that, you had, I'm sorry, that exam that, I was like super worried about at the time, it's just an exam in your entire career. So if you don't ace that one exam, it's totally okay, because, it's the big picture that matters. But when you're first starting as a 21-year-old, starting the PhD in a new place, you want to really prove that you're good, that you're a great candidate, you can do this. You feel like there's so much pressure, and also I think also I felt that it's also the move, right? Because you didn't have the you were not in your comfort zone. You didn't have the support that you had at school or even in undergrad, where you will see your parents over the weekend, your best friends were just right around you, or your roommate was your best friend. So you kind of lack this support system. So that was another layer that you have to overcome. But yeah, I think it was just it's several reasons. One of them is really being passionate about what I do. I really enjoyed what I'm doing. So if I go back, it's like, it was hard. Yes. Did I enjoy it? Very much. Sometimes, some nights were, difficult, but then, it's, as I said, like, it was totally worth it. And I have to admit, it's seeing how hard my parents worked for us to get the education that we got was also another factor, because I knew how much they care about education, and our education to them was the main goal. Like, it was their goal. So they really, we have this idea in mind that education, that's like the best, your best weapon, your best, guide through life. So if you have your degrees, if you have your knowledge, you will be good anywhere. So that's why, like, after seeing how hard they worked for us to do that, I also felt like I can't be quitting, because if I quit, it's also, ruining all their dreams that they worked so hard for. Also having supportive family and friends. So my sister was my biggest, supporter, like always telling me, just let's wait one more week. If she had listened to me and booked that ticket, I would be out of the PhD since the first month. So I'm so glad for the family that I had as well, and the friends.
That's amazing. Yeah, that's amazing. Thank you for sharing. Those more personal things that happened with you. But I want to hold on to another word you said, which is your love, your passion for science, for molecules. I share with you that love. To me, when I was studying organic chemistry and then later biochemistry, oh my God, it's like I was going to this awesome exhibit or this awesome experience just by trying to imagine how these molecules talk to each other, how they move, how they interact, was just pure joy. And I think that is what also kept you, hanging in there, because of that love and passion for nature. And for us, it's peering into the cells and then peering deeper into the molecules, trying to understand how they work. So tell us more about your science, your choice of chemistry, and then your choice of those macromolecules, nucleic acids, those huge molecules that really are the basis of life all over the universe.
Yeah. So during my PhD, I was very excited about the work that was done in Professor Dama's lab at McGill University, where they try to study the DNA structures or the RNA by trying to develop RNA therapeutics, and also study how does we all know we have the DNA double helix, but we also know that, we're trying to understand more that it's not only this double helix that was discovered in the 50s, but there can be other forms. So I was so excited, and like, what are these other forms? How can we study them? How can we stabilize them? And if there are really these other forms, how can they affect us? So the structures I was interested in form at the end of telomeres, which in normal cells, these telomeres shorten with every cell cycle, and then a cell dies, and that's what leads to apoptosis. But then in cancer cells, these telomeres keep elongating, and that's why cancer cells are immortal. So how do these structures differ from one to the other? What makes it get shortened here but elongated here? So that was what I was interested in the Dama lab. And then when I moved to Duke University, I decided to switch from DNA to RNA. And RNA is a totally different world. And it's super, super exciting because especially now with the vaccines, the COVID-19 vaccines, people started to believe more in the power of RNA. And this is amazing to us as RNA scientists. So people think of RNA or as just this one single structure that is forming, some studies from before, like crystallography studies, or when you have a crystal structure when you're drawing it, you're just seeing one snapshot, but that's not true. That's not what it is. So imagine your RNA molecule as a ballet dancer, right? So it's performing, it's moving, it's jiggling and wiggling, it's all over. And then if you take a picture, you're capturing one part of it, one part of the dance, but you might have missed another way more exciting move in the dance. So that's what we're trying to study here in Hashim's lab, to try to capture all of these different moves and see which move is the move that is really affecting the function of this RNA.
Wow, that's amazing. Yeah, that's amazing. You're peering in and following closely these huge macromolecules. Alibet RNA are smaller than DNA, but still they're pretty big as they wiggle and jiggle and move around. And you're still in the early career stage, and that's interesting because we want to know how, not just to interview senior scientists who've progressed in their career, but people like you who are still in the middle, figuring things out and what's going to happen and so on. So that's really amazing because we're going to come back to you, and I want to ask you a question about. Did you ever think of acting out the jiggling and wiggling of your molecule? But we'll talk about that after the break.
Sure.
A free online chat can give you the personalized tips you need to start boosting your retirement savings today.
They took over my bedroom. Come on. Okay, Dad. 1, 2, 3. Dad! You saved me. Dad?
Are you okay?
I'm fine, dear.
Your hero needs you now, and AARP is here to help. Find the care guides you need at aarp.org/caregiving.
Find out how to use an awkward silence to help a friend with their mental health at seizetheawkward.org.
I am what hunger looks like in America.
If I could be you, and you could be me for just 1 hour. Walk a mile in my shoes. Walk a mile in my shoes.
Right here, baby. Look. Uma.
Noah. Something more than a birthday is happening here. Once you can see it, you can help. The sooner you recognize the signs of autism, the sooner you can make a lifetime of difference for your child. Start by answering a few simple questions at screenforautism.org.
Welcome back to Science Connections. We have with us today Dr. Hala Abou Assi, who's going to tell us about the jiggling and wiggling of RNA, one of the most fascinating molecules. And everybody's, hearing of them more and more today because of the COVID vaccine that was made, especially the Pfizer one, based on this new technology of RNA. So tell us, Hala, about the jiggling and wiggling. And have you ever thought of acting it out in the lab, you and your colleagues?
Actually, some of my colleagues did go to a studio and dance, and another colleague took like a picture of them that showed the motion while they were moving. So it's not you're capturing one picture, but it was like a collection. So and actually that these pictures that they took one in the RNA Society conference talent. I think it was in the talent section where you send like pictures you've taken or anything you've done related to RNA, and it did win because it was super relevant, and that was super creative of them. But two of them were actually ballet dancers. So when this idea like came to their minds, it was really amazing how they executed it. And maybe later I can share this picture with you. So yeah, they did actually do it. I didn't, but my friends did it.
That's amazing. That's amazing, because that also is an example of the interdisciplinarity of scientists. And if we're going to go ahead and solve the problems of the 21st century, we need all the disciplines to come together. And most importantly, we need to portray how science works to the general public. And one way is integrating science with art. And art has many forms, and one of them is dancing. So this choreography that you're talking about is fascinating in order to share with the general public a better understanding of the molecules you study. So that's amazing. So tell us more now about your actual research with these molecules after you dance them out. What does that mean? What does your research mean? And what are the challenges in the lab that you faced as a scientific researcher, designing experiments, running into problems, which I know we all do, and then how you deal with those? I don't like to call them failures. I like to call them opportunities for learning, and what keeps you going inside the lab.
Sure. So the lab here studies, as I said, or develops techniques in order to be able to study this dance, because using like some conventional techniques, it's very hard to be able to see these RNAs and their different performances. So in order to be able to see, because either they are very fast. So if you have a super hard move in your dance, it's probably going to be a super fast one so that you don't get exhausted, or if it is super low and populated, so there isn't much of it, so it's hard to see it when there's not much of it or spot it. So that's why in Hashim and Hashimi's lab here, he is the pioneer in developing a technique, NMR techniques, nuclear magnetic resonance techniques, in order to be able to visualize these very low populated and short-lived states of this RNA. So when I joined the lab, we were interested in how do certain modifications of this RNA affect its jiggling and wiggling. So if this dancer has a certain, let's say, if she's wearing certain outfit, or if she has a certain injury in her toe, how will this affect the dance? Will it affect it in a way that it's visible using these advanced techniques that were developed in the lab? And then if we can, I'm work in two labs. One is in Professor Hashim al-Hashimi's lab, which is more biophysical, more hardcore NMR, in order to be able to see these molecules. So it's more on the structural side. But I'm also co-supervised with Dr. Christopher Hawley in the Department of Medicine, where it's more molecular biology. So when we know there is a certain molecule decorating RNA, and we know how is this molecule introduced to the RNA, I can go to the other lab and then be able to remove the cause of this modification, which we call either like knocking out or knocking down the enzyme that leads to a certain modification of an RNA, and then see, okay, how did this affect a certain functional outcome or biological outcome? So if I remove a certain modification on an RNA that is affecting how our mRNA is spliced to, then will it affect splicing afterwards? Maybe I'm getting into too many technical terms. But in general, it's the power of understanding how can a certain decoration on an RNA that can be super small affect its overall dance and then overall outcome, as in function, like will it affect a certain pathway, biological pathway. So
wow, so science is all about teamwork, right?
Exactly. Yeah.
You can't do science, it's not a one-man show anymore. It has to be a number of scientists working together, each coming from their own discipline to help solve the challenges, to push science forward. And it's
Absolutely.
also standing on the shoulders of the people before us, right? In terms of who developed the NMR technology and others before us. And then we provide the basis for future scientists to come before, which to me reminds me how sometimes when we're doing basic science, people think, oh, this is not important. Who cares if you're studying, because you're really studying just one little molecule and focusing everything on it. But really it matters on the long run because you're adding a piece to the bigger puzzle. And maybe in a few years, that piece will be very, very important when today it may not seem to be. So could you share with us a little bit more detail, if you want to give us an example of a research experiment you're doing, or a paper you've published, or a discovery that you've made, whether you or within the lab you're working with?
Sure. So yes, definitely. It's basic science is definitely needed. It's the foundation. And yeah, so for example, in my work, we were able to show that a very small, tiny addition to the RNA molecule can affect how it is forming and favor one formation or increase the population of one structure, which or one conformation, which was not that present before. So, and yeah, just imagine your RNA. So the RNA, or the different conformations of the RNA, some people think that it is just forming it's the same thing, it's the same molecule, it's just under different conditions. So when you have this music and the ballet dancer is, performing, she will or he will, respond to the music and move based on the different music that's being played. So that's exactly how the RNA is behaving. So whenever you have a different cellular condition, stress, pH, temperature, or these decorations that I work with, this RNA will respond differently and just fold differently. And it is now we know that the functionally relevant conformations of these RNAs are usually the ones that are hidden, that we don't see when we run conventional techniques. And one quote, which I always say, in when I am presenting my work, which was written by Dr. John Stites. She was referring to RNA-RNA interactions, but I use it when I'm talking about our studies and about RNA dynamics. It is when you don't think of RNA like this dancer, and that has different conformations based on the different stimuli. The quote says, ignoring this landscape of RNA is like walking blindfolded into the beautiful wilderness. And that is totally, in my opinion, when I read that, I'm like, that is super relevant to our work, because that is the case. If you just ignore and don't think of RNA as this molecule that can very much have different conformations, you can't actually be solving the problem. You can't target it, because now we know that the functionally active or the functionally important conformations are the low populated in most cases. So when you are able to see how these low populated conformations are, now you can target them. Now you can design small molecule drugs to target them and solve the problem early on before it progresses afterwards.
Wow. I mean, I love that quote. Like if we don't understand the landscape of RNA, it's like walking blindly in the beautiful wilderness. Amazing, amazing. And that's the beauty of nature, right? Just to realize about this beautiful wildness and wilderness around us. And we have the opportunity to go in and look around and poke around. Oh my God, that's what keeps us up every day, right? That's what makes us go into the lab, even if we encounter failures and experiments that don't work. So my question to you, when something doesn't work, you plan the experiment and it doesn't work, and that happens a lot. What do you do? How do you deal with it? Do you go for a walk? Do you go read? Do you say, I'm going to leave this for a while and come back? What do you do?
Yeah. So, I mean, science is the case. When, the experiments that didn't work during my PhD or even now are way more than the ones that worked. But then that's what makes you more knowledgeable, because when an experiment didn't work, you have to go and figure out, why did this happen? Why didn't you see the product that you were aiming for or the result that you wanted? So it will require you to think more and to problem solve, and then that's what builds your knowledge. So I'm of course, like, day by day when something doesn't work, and to me, because I'm so passionate about the work that we do, and I was very lucky to be in labs where I really enjoyed the projects, and I feel like they're part of me. So when something doesn't work, I feel like like your baby is sick or there's something wrong. So it's not like when I leave the lab, it's like I don't care anymore. No, it's always on my mind, and I really, really want to. So I feel like I get so sad, or I used to get so sad before, and now I feel like I want to go back the second day and as soon as I can to figure out why this happened. Of course, if this requires some reading, I will have to do the reading. If this requires to order new stuff, I will have to order the new compounds to try something new. But yeah, it's always like trying to get back and figure out why. I think this was very. To me, that was what I have always done. I have to admit, I always got sad when, this happened. And, I even when I call my parents or my, my sister, my husband, I'm like, this experiment today didn't work. I'm like, it's like, so, everyone has to know that, there's something wrong with my reaction, because that's how much it meant to me. But yeah, you learn from, what doesn't work and make it work to have a beautiful story at that. So
exactly. I mean, that reminds me, when I'm in my lab doing my work and the experiment doesn't work and you feel upset, of course, but going back and being with my kids and family, it's a good break. You freshen up, you think of other things in the world, and then you come back fresh and with new ideas and a new angle to look at, a new perspective. So actually this going back and forth between different contexts is really, really very innovative and allows you to think outside the box. But we keep going, because we tell ourselves, what got me into this? The experiments are not working, but as soon as one of them works, you forget everything. You're over the moon. It's like when you have a baby, you forget everything. This is the most wonderful baby ever. Exactly. I love your example of a baby. Well, We're going to be back to learn more about how your science impacts our everyday lives. So stay tuned.
A free online chat can give you the personalized tips you need to start boosting your retirement savings today.
If I could be you, and you could be me for just 1 hour. If we could find a way to get inside each other's minds. Walk a mile in my shoes. Walk a mile in my shoes. Well, before you abuse, criticize and accuse. Walk a mile in my shoes.
Dad, they took over my bedroom. Come on, come on. Okay, Dad. 1, 2, 3.
you okay?
I'm fine, dear.
Your hero needs you now, and AARP is here to help. Find the care guides you need at aarp.org/caregiving.
Find out how to use an awkward silence to help a friend with their mental health at seizetheawkward.org.
Right here, baby. Look. Uma.
Something more than a birthday is happening here. Once you can see it, you can help. The sooner you recognize the signs of autism, the sooner you can make a lifetime of difference for your child. Start by answering a few simple questions at screenforautism.org.
Welcome back, everyone. We're so excited to learn more about the jiggling and wiggling molecules that Dr. Hala works with. Dr. Hala, we want to know from you, how and in what way do the molecules that you work with impact our everyday life? How does RNA play a role in my life when I wake up in the morning or go about my day?
Sure. So I'm happy that we're having this conversation after the vaccines came out, because now people believe more in the power of RNA, because before it was hard to convince people that. So I will give an example, which actually it's a title that my previous supervisor used for one of his talks, which is turning off the tap or mopping up the floor. So what we're trying to do is we're trying to understand how this RNA is forming into different conformations. What conformation is the one that is causing a certain disease? And then we want to target this RNA with a certain drug. The most drugs that we have now are mainly target the proteins. So in life, we have DNA, which is then DNA, and then RNA, and then the RNA is translated into protein. And most of the drugs target this protein. So if we can come and design drugs that can target the RNA, then we're solving the problem early on. So that is what my supervisor meant by turning off the tap, instead of waiting for the protein to form, and then there's this disease, and then you will target this disease by targeting the protein. So, but to get people to visualize RNA as something that is druggable was hard. But now with so much advancements in techniques and in research done in our lab and in other labs, we are now having the power to be able to, to get closer to our goal, which is to, to design drugs that will bind a specific RNA, a specific conformation of an RNA, and solve the problem instead of waiting so long for it. So yeah.
Can you give us an example of a drug that targets RNA that's in the market?
So
or that's not in the market yet. The RNA drugs are not yet in the market. This is too early in the pipeline of drugs.
There are RNAs. So one side of it is RNA, designing RNAs that can be drugs. So that's already in the market now. There's a few of them. But yeah,
so the RNA that are used as drugs are used with what diseases?
Yeah, so I know there's one for high cholesterol. There's one for some angular muscular dystrophy. Yeah, so these are the RNA therapeutics. But yeah, I can't recall the name of a drug now that, yeah.
That's fine. So this is very early, right? This is really early stages of using RNA, whether as a drug itself or use it as a target for a drug as well. So you can look at it in different ways. So how does that relate to the vaccine? Because we know that the vaccine is based also on RNA. So could you explain maybe a little bit in simple terms, what is the role of RNA in vaccine design and making it? production.
Sure. So when you know what the disease is, what the sequence of, because RNA or DNA is a certain sequence. So you have certain building blocks. There are 4 building blocks, and they are in a certain order, right? So when now we have the ability with all the sequencing techniques, with all the advancements in science, to know what this sequence is. So if I know what this sequence is, I can design a certain RNA that will go and bind to this RNA, that my target RNA, and by sequence complementarity. So I know exactly where it's gonna bind because of the complementarity that we have. So some say there might be off-target effects that can go by otherwise, but studies are done and are really like accurate. So to make sure to limit any off-target effects that can occur. So you design the certain RNA, and then in addition to designing the RNA for it to go and target another RNA, let's say, there's another challenge, which also people are working a lot on, which is delivery. So when you design an RNA, that's not something that I personally work on, but that's a challenge in the field, which is you need to deliver, or like you have this RNA, you need to let it go to the specific cell that you want it to destroy or to target or to cure. So and that is a very also important field in RNA therapeutics, which is the delivery. So to make sure that you have the RNA packaged in the right way to be delivered safely to the right cell to do its job. So RNA is really a big field where, as you mentioned, you can have RNA itself being a therapeutic. You can target it, and then for it to be a therapeutic, it has to be delivered, packaged nicely, carefully, to be delivered to the right place.
So it's multiple stages, from basic science to understand how these molecules dance, jiggle and wiggle, and how they form, and then to understand their function, in cellular function, how they affect the cell, so that it's a normal cell, whether it's a liver cell or a pancreatic cell or a neuron cell in our brains or a muscle cell. And then after we understand the normal biology, if something goes wrong, we want to know how to fix it. And so we want to know if RNA plays a role in what's going on that's wrong, and then try to figure out how to fix it. So it's multiple stages. And then after you understand all this, you need to think of delivering or designing a drug, and then deliver it, and then send it in. So it's a long process, and usually it takes time. But what's beautiful is how when we work together, we can make things happen faster. And so I think one success during COVID was because of all the past research we had before, we were able to accelerate, because all the world needed this vaccine. So all the world scientists put their heads together and worked hard so that we could produce this vaccine that could be effective. And by the way, this is not just a job that's done. It's still ongoing because we have new variants, and therefore we have to keep on developing new drugs. And that's why science is so exciting, because there's always something new we want to discover and some human we want to help. Because ultimately, as scientists, our goal is to help humanity. And that's what keeps us also going. That's not just, playing around in the lab, that ultimately our experiments will have an impact. So tell us, Hala, if you were to advise yourself at 16, what advice would you give yourself back then? And expand it maybe to advise the young generation today, because, time flies and times change. And when we were teenagers, when you were 16, when I was 16, way before you, I don't know how much we are similar to the 16-year-olds today, or maybe we are.
Sure. So some advice I can give from my personal experience is to really follow your dream. Don't let, which probably this is cliche, but that's true. Like, don't let, what others say that you cannot do it, or it's hard, or even you, when you say it's more with you. I think I said that something was hard more than anyone said it to me. So, don't let this affect your dream. Find a passion. Make sure you don't be forced into a major or a career. When you are in a field that you like, you will go to work every day excited about it. So find find this domain that you will wake up every day excited to what's going to come, whether it's a failed experiment or it's a win experiment. You will still be excited, because at the end of the day, it's the outcome at the end, not just day by day. So yes, follow your dream, find something you like. And I think another example, another advice which I will give, is to make sure to find or pick your mentors wisely. Find mentors that care, will give you mentorship, not only probably about your experiment or your research, but also about you, because each person in the lab is different. So the mentor should really be giving mentorship, not limited to just research. And also the mentorship doesn't end with when you graduate or when you leave the lab after a postdoc. And I have been so lucky with the mentors that I had. My undergraduate, PhD, my postdoc supervisors, they have been amazing. And I think I'm speechless. I don't know how to thank them, because they definitely were amazing. They genuinely care about my work, but also about my life, my family, now my small family, my baby, which have helped me so much, because you connect at a personal level, not only a job. So I found that very helpful throughout my career, when you're working with mentors that care and want you to be your best, and want try to push you to be the best version of yourself. I think this is instrumental for your career and life. So yeah, if you can, make sure to pick mentors wisely.
Well, you mentioned you have a baby. Wow, that's amazing.
Yeah.
Tell us more, because I have 4 children myself, and I'm a scientist, and to me, you could never separate both. They're all integrated together in your life. You love your science and you love your children, and you want to succeed in both. They're both your projects. They're both a gift to humanity. So if you want to tell us a little bit about how you enjoy being a mother and a scientist at the same time.
Sure. So my daughter her name is Mia. She will be 1 year old in a couple of weeks. So yeah, I think her presence changed my perspective in several things. So now you know now what matters more and what you should really be worried about and what not. So if an experiment doesn't work today, before I would go home, stress so much about it, try to keep thinking about it. Now it's like I disconnect, I go to my baby, to my family. We, it's, we have fun. We try. Now she's like, she has her own experiments, try to explore, try to walk, So yeah, I think like this connection that was naturally happened, because of course when you go home and you have your baby, you're not going to go to your office and start, going back to work. And I found it very healthy, because now I have like my work, I have my baby, it's my family. So I think now, although I was so worried before, like, is it the right time to have a baby? I think she is the best. Yeah, definitely life changed so much after she was born. And yeah.
That's amazing.
She has a little RNA, she has a little RNA for babies book, which my supervisor gave it to her. So
that's
yeah,
amazing. Does she have Lego RNA, little models that she plays with?
Yeah, well, when she's a little older, I will get her one, because now I'm worried she will, swallow one of the pieces. But I did get my nephews one of the, these, like, not Lego, but like, it's magnetic. So we built a DNA together. So yeah.
That's amazing. I think, whether someone is a scientist doing experiments, contributing to science, or being a parent, whether it's a mother or a dad, taking care of the next generation and bringing them up to be healthy and curious with critical thinking skills, both are very, very important. And we cannot survive as a species without having both. We need both. And that reminds me of a famous saying that says, what are humans? They're seeing what everybody sees, but thinking what no one has thought, right? Thinking that curiosity, that innate curiosity that drives us, that we need to foster in the young generation growing up. So I'm going to end by asking you, what's next for you, Hala? Where are you planning? What are your plans? What are your dreams?
So currently, I'm at a very actually critical stage where I want to decide what's next. So I'm trying to find what will be like the best position after to be able to contribute to do the science that I really enjoy and try to have more direct impact. I have been now like thinking about some companies where to join, but I don't have the decision yet. It's an exciting week because I will be deciding sometime this week. So wish me luck.
That's amazing. So you're at the crossroads of whether to continue in academia, find a position in a university, or to continue in industry to contribute to drug design and drug delivery. That's a great place to be, and we will follow you. So we will follow you, and we look forward to learning more about your discoveries and how you've impacted science and humanity, whether in your lab or at home with your baby, with Mia. Well, thank you very much. Folks, stay tuned to our next episode when we will be interviewing a very interesting scientist who works at MIT.